Yeah, I was trying to do a recording for a previous class and ended up having to do it 3 times because forgot to share my screen the initial time. All right, so let's pick up where we left off with our chapter 9 discussion of the techniques that are involved with nucleic acids. All right, we're going to look at eukaryotic gene expression in bacteria, and maybe a little bit in eukaryotes. But what we're really trying to do here is take a eukaryotic gene and get bacteria to make it. So the general machinery inside the cell is similar, but we can't do a lot of the modifications. When you think of your eukaryotic genes are a little more complex, the proteins are maybe a little more complex. So you can have some issues, but there are workarounds in here. Okay. So, our eukaryotic gene typically won't express correctly. So, because our eukaryotic genes have introns and exons, and the exons are coding regions, and the introns are non coding regions that are spliced out during the construction of the mature mRNA. Okay. I guess that'd be the mmRNA. Yeah. Um, so introns can pose problems for bacteria because they can't splice them out. Little asterisk there. There are a very tiny number of bacteria that are capable of dealing with introns that were recently discovered to actually have introns of their own in there. So maybe there's another series of workarounds using this particular type of bacteria in the future. But right now, with the way that we commonly do this, stop. Sorry. Oh, come on, man. Let's get back here to where it was. Nothing like the trackpad. Oh, no, too far. Going to give everybody seizures here. All right. But anyway, so bacteria typically can't splice introns out of the coding DNA, so we're going to work around that. All right. So what we're going to do is use a constructed DNA in there, or a cDNA. Our mRNA is intron free, so our mRNA already has the codon spliced out, so we can use it as a template to make our. I'm getting ahead of myself. To make a DNA sequence, and then transfect that DNA sequence, that constructed DNA, cDNA, into the bacteria to get the bacteria to express the mRNA and then make the protein. All right. So that's something we have to do to get bacteria to express eukaryotic proteins. All right. So a few definitions to start off with. Eukaryotes are uniformly monocistronic. Basically, one gene gives us one protein. Okay. Bacteria are quite often polycistronic, where you have one segment of RNA or DNA, and you have multiple genes in there that will all be expressed. Okay, and that can be an efficiency thing. Bacteria are small. Don't have a lot of stuff going on. So if you can compact several genes that are usually related to each other in their function into one series, then maybe you can get something going there. All right. I don't know why my trackpad is being crazy today. We're going to just not use it. All right. So how do we construct our DNA, our cDNAs here? We can extract mRNA. Again, it's really fragile, so we have to be careful about that and have to make sure there are no RNases around. And it's really a great deal of care in the procedure that's in there, but we can extract mRNA. The neat thing is all mRNA molecules will have this poly A tail. All right, so we can purify them very easily. You have beads with, say, like a poly T on there, and I'll just pull them away. Nice universal template for primers as well. But we can basically synthesize a complementary DNA strand using the mRNA as a template. So we'll utilize reverse transcriptase. Reverse transcriptase we talk about a fair amount in Biochem 2. But we see this as basically something that can take mRNA as a template and synthesize a complementary DNA to it. many viruses will utilize reverse transcriptase to help integrate themselves into the host cell. HIV is particularly pernicious in this regard. There are also, with the discovery of reverse transcriptases, that is another avenue for possible gene therapy, so that you can cut out a defective gene and ideally replace it with a new one by using reverse transcriptase to insert that into the host chromosome. Anyway, for our purposes, though, we're just going to simply take that mRNA as a template and make a DNA strand in there. Um, so now we have this hybrid DNA RNA double-stranded nucleic acid. We could degrade the RNA very easily while maintaining the DNA and synthesize a second complementary strand that would have the same sequence as the RNA, except with T's instead of U's, and make our double-stranded DNA that way. Right? So this then becomes our constructed DNA, our cDNA. Okay. So to go over this methodology, come on, there we go. Yeah, I'm a little worried here about my computer. Okay, so here's our RNA with our poly A tail. Here's our initial primer. We start this to bind here, our reverse transcriptase. We'll start with our multi-T section, and then we'll synthesize whatever this complementary strand is. So we have our green RNA and our blue DNA. Okay. Our next step, once we have this double-stranded device, we treat this with some RNase. RNase will degrade the RNA, but leave behind the DNA. And then, if we know our sequence up here for our DNA, we can add another primer here on the 5' and 3', and fill in whatever our original sequence was to make our double-stranded DNA. And we now have a cDNA that mirrors the sequence of that mRNA. So that would be a DNA strand that expresses the matured gene for for whatever gene of interest that we have. Okay. This is important if we want to actually study the protein products. And again, I'm a protein biochemist, so all that, yeah, we're gonna put a gene in here, and yeah, look at that. It was a very interesting split when I went to grad school, because I could choose, I can go either into the gene and nucleic acid field, or I could have gone into proteins, and I like proteins so much better. So if we're going to copy DNA, we have we talked about cloning DNA previously. Okay, so now we can talk about not only just cloning the DNA, but expressing the protein from that gene product in here. So we're going to use expression vectors, which are very similar to our cloning vectors, but just have some extra stuff in there. Okay, they allow us to transcribe this gene. Okay, so that means they're going to need to have promoter sequences and operator sequences, and a ribosomal binding site code, as well as a termination sequence in there as well too. So let's take a look at this. All right, so here's our origin, just like our origin replication, just like our standard cloning vector. But for our expression, we have to have these promoter and operator sequences in here, our polylinker, our ribosome binding site, our termination site, and of course, just as before, we have our genetic marker resistance. Okay. Alrighty, and then we may have some other stuff in here that helps regulate this. So expression vectors can get, pretty full of extra stuff. Just like we can keep adding stuff to our cloning vectors, but these are the bare minimum things that we need to have. All right, in here. Alrighty. So how do we purify them? So natural protein purification can be difficult. All right, but if we're making a constructed DNA, we can make it easier. We can tag them for purification. Okay, so we can use a lot of different tags in here. We can generate ones that will bind particularly to a resin in a column or in beads. So we might have like this protein A. We might have a series of histidines in here that will bind to nickel. This is probably the least invasive because it's really just 6 histidine residues that you stick on the end, and they will bind to nickel, and it makes it very easy to add on there. And ideally, it will have very little effect on the overall structure. Here we're adding, significant molecular mass, so you might see some, might have unintended effects. We'll talk about that a little bit. Glutathione is another one, maltose binding protein. Beta-galactosidase was one of the ones first used. You can even have like a chitin binding domain to bind to chitin in there as well too. So that's kind of a neat thing to pull from the insect world. Bind to chitin because you don't see that too often in eukaryotes. Well, complex. I take that back. Take that back. Where's chitin in eukaryotes? But yeah, not usually internally. Definitely don't see that in bacteria. Um, all right. But the point is, we can find some place on the protein that ideally will not affect the folding of the protein, that we just attach something extra that makes it easier to purify. We can also do this, we'll talk a little bit later, with making it easier to visualize as well too. We have a whole batch of fluorescent proteins, starting from the discovery of green fluorescent protein. We could add that to these as well too, to allow us to see where the proteins are localized as well. So, when we make our recombinant genes, we can add stuff that makes it easier for us to do whatever it is we want to do, whether it's purify it or figure it out. As far as the purification, okay, our gene is in red. Sorry, yes, yeah, and then we add this glutathione gene in there as well too, and attach those together so that we essentially get our particular gene product that is fused to glutathione. Okay, so we have this fusion protein, and it gets expressed in the cell. We can take, we can chop up our cells, make our whole cell extract here, and pass it through a column, and our glutathione will bind to its glutathione binding partner in there, and everything else should wash through. Why is this not? There we go. Ooh, this is disturbing. Anyway, our glutathione tag will bind here, and everything else should wash through. We can then elute, maybe with a high salt solution, or more glutathione or whatever in here, glutathione solution, and we can ideally in the pretty much one to two steps pull off a purified protein. Okay. Um, I mentioned before, I believe, that we don't have to use columns anymore. I did a purification where it involved magnetic beads, and the beads were essentially the same thing as the stable phase, I guess the stationary phase in there. These beads were essentially the resin in the columns, and you could just put them in a small tube and then incubate that with your whole cell extract. Usually, you kind of have it on a rotating, I don't want to say incubator, what's the word, but basically just allow it to mix, and so that the protein of interest with its tag would bind to those beads. Then we'd spin those beads down to the bottom and aspirate off the rest of the solution in there. Add in an elution buffer. Let that incubate for a couple minutes. Pulls off the tagged protein from the beads. Spin the beads down. Then when we aspirate out the supernatant, we should have a pure solution of our protein. And rather than column can take a couple hours, I was actually able to purify proteins using that bead protocol in like 27 minutes. So yay speedy time. All right. So besides being able to tag them for purification, we can tag them for location. I mentioned green fluorescent protein. There's also BFP, YFP, RFP, red, blue, yellow, cyan, orange. There is a whole raft of options of fluorescent proteins now that we can utilize here. So instead of instead of maybe tagging with GST, we tag it with GFP instead, and then we allow it to be expressed, and we can figure out where it is. Okay, so we can use a fluorescent microscope to figure it out. So we can also do immunofluorescence as well too. We can tag the protein with a primary antibody, okay, and then we can detect it with a secondary antibody that has this tag on here. We could also, instead of using the primary antibody, we could just use some short epitope that we have a primary antibody to, and we can detect. So here we can see this is the structure of our GFP in here. We have this nice little beta barrel structure with a fluorophore that's kind of trapped inside. By making alterations to this, and that's actually one of the kind of the neat things about one of the links. I think it's the link that I linked to you, that you can make um, single point mutations that enhance the stability or enhance the detectability of these. And now we have something like 14 various fluorescent proteins of varying colors in here. So if you want to see if a bunch of proteins interact with each other, you could tag something like 14 different proteins. Okay, some of these colors are pretty similar, but you can tag at least a half a dozen and see where they all are in respect to each other. Okay. So for example, here we have a nematode. Pay no attention to the purple in there. It's just kind of something, but we can look at the green fluorescent protein showing up at a particular neural sequences in here. What we have here we have our plasmids. Oh, come on. What a pain in the butt. All right. Anyway, these various tags can help us figure out where stuff is. Okay. And again, our fluorescence proteins in here, we can look at, say DNA polymerase A. We're looking at a couple different components of that, and then we can do this merge of our our GFP and our blue, in this case BARDU, I guess, whatever we have here. But if we merge that and we can see the color overlap, we kind of false color that as yellow and say, hey, here's an interaction that's saying that these two particular proteins are going to co-localize. Okay, and usually that means that they're co-localizing, there's an interaction between the two. So it's kind of like a fishing interaction. Hey, does this protein interact with another protein? We can tag both those proteins and then see if our fluorescence overlaps. Okay. So again, looking at this in kind of a summary, we have a promoter sequence in here, our cDNA, our particular gene that we're interested in, and our green fluorescence protein. I believe this is the sensory neurons of a nematode. So they're the ones that kind of allow it to feel its way through the earth as it's moving around there. So they are, of course, localized to the surface of the nematode. And you've got a couple that go across in here, probably to its, connecting to its brain in a way. Nematodes, very simple, very small brains, much like professors, right? All right. What else we have here? Okay. So I sort of mentioned this with our overlap, but look, how do we figure out if two proteins are interacting with each other? For example, if we look at the electron transport chain, complex 3 and complex 4 are right beside each other, and they interact to kind of shuttle electrons through the electron transport chain. So how do we figure this out? Well, we can isolate this protein complex. We can tag one with an epitope, and then if we isolate that, hopefully it will pull down the other proteins as well too. Okay, if we had to have very gentle separation procedures for this, so not a lot of harsh detergents that'll separate them out, but once we pull down everything together, we can be more harsh and separate them and try and figure out what's in there. We can also use these tap tags that will allow us to enhance this. So this will give us 2 purification steps. So we can pull down the whole complex, and then we can eliminate loosely associated proteins and get rid of any non-specific binding that shows up in there. Okay, so our classical method is something like this. Pull down here, we get our pure tag protein and our immunoprecipitate. These will be things that are associated with that. Our tap tag, we have, by virtue of, again, our promoter, our cDNA, and our tag in here, we have this tap protein in here. So we can see that we can pull down a very nice gentle isolation in here, and then we can cleave off part of this tag here as well. And then our more tightly bound proteins, we can isolate over here as well. Okay, so we can figure out what our eluted protein complex is. So we can figure out loosely associated and tightly associated proteins through this dual process by virtue of having a nice little cleavage site in here that allows us to cleave off, in this case, our protein A. So in essence, what we're really doing is we're tagging our cDNA with a couple of particular epitopes or reporter genes in here. We can call this whole type of deal we can call reporter genes, that they basically allow us to show where it is. I always think of like the youngest child saying, "Hey, the older child is doing this. He's getting in the candy. He's sneaking out the window. These are our reporters. They just kind of tell us where they are and what they're interacting with." All right. There's also a yeast 2 hybrid system. Okay. Our yeast 2 hybrid system relies upon our protein of interest being tagged with a Gal4 activation domain. Okay, so basically the reporter gene ends up being under the control of Gal4. So if we have Gal4 in our plates that we're growing them on. I don't know if you guys can hear my family yelling in the background here. Like, don't get popsicles. I swear, need a white noise generator. Anyway, under the control of Gal4. So if we express Gal4, then we will express our proteins. If we don't have Gal4 in there, then we won't get them in there. So we have to, these differentially tagged proteins have to interact with each other in order to get this to express, okay? So we have our yeast strain that has a Gal4 binding domain, our yeast strain that has a fusion domain. The two of them have to interact, and then we'll get these surviving colonies that will form up here. And I believe we can actually make it turn blue as a reporter assay as well too. All right. Anyway, not the most exciting of these reporter systems in here. I don't like the yeast 2 hybrid system personally, but that's because everything I dealt with, I could grow up in bacteria. So, and I actually dealt with yeast. All right, come on. Okay. All right. Let's switch gears and talk about microarrays a little bit. Our microarrays or microarray chips contain fragments from genes to be analyzed. So we basically take an entire gene and we chop it up into a whole bunch of different pieces. All right, and then we can figure out what pieces are where. And we can also take pieces of the entire genome of bacteria or yeast and fit them on a single chip. We're getting close to doing that with, like, say, higher eukaryotes, but we have these differentially tagged either mRNA or cDNA pieces in here, and then we can analyze the chip. Take an extract, pass it over the chip, see what binds there, and we can show differences in expression patterns. All right, so how do we do this? What we want is we want different sequences on our little plate here. So we're going to take just a little 4 sequence plate here, and you can have thousands of these on a chip, okay? So what we do is, we want to start with adenine at positions 1 and 3, but not 2 and 4. So we block these sites here. We allow these to have light in them, and then we treat with this photo label adenine, and that will bind to our surface here, okay? But not to the ones in the dark, okay? So then, next we want to add guanine, all right? So we don't add guanine to either of our adenines in here, so we make sure they're in the dark, but we add our photo-labeled guanines in here, and they will both bind to positions 2 and 4, okay? So now next we want to add C, all right? What's going on next? Yeah, but we only want to add C to position 4 to the guanine that's there, so we cover up positions 1, 2, and 3, and then add our treated cytidine in there, and it will bind there as well too. Next, you can see we will be adding thymine to positions 1, 2, and 3. So we cover up 4 and do that, and we just do this over and over again. I got this down to a robotic sequence in here, but the goal is we can kind of build up these sequences in different spots, okay? And we do this at hundreds and hundreds, if not thousands, of little microdots in here. So we have these little tiny areas where we have a particular sequence, okay? And that sequence will be complementary to either an mRNA or a cDNA, okay? So one way that we can do this, okay, is we can take, in this case, we're looking at maybe this is not evolutionary, a developmental assay in here, okay? So we may take a fertilized egg, right, and tadpole, isolate mRNA from both of those, convert that to cDNAs, okay? And then we take our 2 different cDNAs and we pass them over the microarray here. This one might be labeled with GFP and this one might be labeled with RFP. And we can see where they bind will tell us what genes are being expressed, okay? Because we have complementary sequences here, right? So we'll separate these, or denature these and allow them to bind, and where they bind. So the green ones will say, hey, these are genes that are being expressed in the fertilized egg. The red ones will be genes that are being expressed in the tadpole, and the ones that are yellow, okay, are being expressed in both places. They're genes that are expressed both in fertilized egg and tadpole. So things like S100 or various housekeeping genes, they're kind of always on. We can kind of utilize those as a control as well to kind of figure out where stuff is and maybe quantify as well. But we can also see what genes get turned on and turned off in different stages of development. We can also compare different organs this way as well too. So maybe look at kidney and liver extracts and see what genes are similar between the two, and what genes are different between them. Okay, so there are tons of applications for microarrays. Um, One of the neat things is because you can have these chips that will have thousands of different sequences on them, you can do high throughput screening. So you can look for what genes are present in an individual. And this is where our medicine is moving towards risk factors. What genes are implicated in risk factors? You may have heard Angelina Jolie had a preventative mastectomy because of a family history and her genes expressing BRCA2, BRCA2, which was implicated in breast cancer. More recent research suggests that BRCA2 is not the you're definitely going to get breast cancer that it was thought to be 5 or 10 years ago. So preventive mastectomy may not have been necessary, but we're continually learning more in here. Um, but so we can kind of do gene typing on individuals to say what are your risk factors for different diseases. Are you at risk for Parkinson's? Are you at risk for Alzheimer's? Let's figure cardiac disease. Let's figure all this out. Again, our specific tissue specific gene expression. We have proteins that do the same thing but are slightly different in their sequence or in their regulatory abilities in different organs, to kind of help differentiate the function of those organs. So we can look at tissue specific gene expression to see like, hey, do we have this glucose transporter, or do we have a variant glucose transporter, something like that. We can also look at mutational analysis too. The interactions are sensitive enough that we can often tell whether a particular sequence has been phosphorylated or dephosphorylated. So we can look at that, and if we look at mutational analysis, we may see different genes that have been mutated as well, and that can help us learn more and more stuff. There's a ton of things you can do with this information, and it's hard to express all of it without making an entire class on microarrays. There are, that would be like a grad level, an advanced grad level course, but you could do microarray application in there. Okay. And we'll see, we'll refer back to this a couple times in here. It's increasingly common and used a lot, and simplifies things. The biggest thing is high throughput screening. You can test a lot of stuff at one time. All right. It's kind of like we don't do restriction enzyme digests anymore. We just do sequencing. Okay. So speaking of sequencing, we have a new generation of DNA sequence analysis too. So we can, again, fragment the full genome, just cut them up into a few hundred bases. I always think of like, if you have like a small child eating spaghetti, I'll cut up the noodles in small piece and bite-sized pieces so they won't, try and chew the whole long thing at once. Makes it easier. I tend to be a straightforward thinker. So I kind of like the genome. It's like, oh, I'll start here at this end and go all the way to this end. But we just don't have the technology to consistently, accurately, completely cover that. So we cut it up into small fragments, we sequence those fragments, and then we put those fragments back together in the correct order. So that would be our chip sequencing technique in there. We have pyrosequencing. We synthesize the DNA from the template. We only make one nucleotide available at a time. It's kind of nice that we only have 4, but we add them with a pulse. We can basically read 400 or 500 nucleotides at a time this way. We also have something called reversible terminator sequencing, where we can add a fluorescent tag to the sequence and detect it. And then as we remove that, we extend the sequence and we keep kind of going on to detect that way. Okay. So if we look at pyrosequencing, I'm not going to go into any great detail about this. I might just generally give you some questions about, what are some ways that we sequence DNA here. But our pyrosequencing in, again, we're pulsing in, hey, what will bind here? Okay, nothing binds here, so there's no flash of light. So we don't add adenine to the sequence. So now I make guanine available, and then when guanine binds to the cytosine here, we get the pyrophosphate released. We get our concomitant flash of light as it interacts with its detecting molecule. So I use it in luciferase and we can say, hey, here's an adenine in this sequence. And then you can see here our sequences, 2 guanines. So we'll see a twice as intense flash of light because we had guanine in 2 places. And we see, a thymine and gets added here, and a cytosine And we add our adenine next. Here, there's no flashlight, nothing happens, so that's not part of our sequence, but guanine is next. So we just figure out where these flashes are, and we can read our sequence that way. So our sequence is going to be A, G, G, T, C, G, T, T. You can see we've got 2 here, so on and so forth. If we added like 10 in a row, we would have a wave intensity 10 times as high as just 1. So that's the way that our pyrosequencing works. Not exactly easy to see on here, but there's a way to read this across this way. And I did do some sequencing DNA gels at one point. It was really fascinating, but these were big gels. This is a much better way. Our reversible terminator sequencing, again, is very similar. We just kind of figure out where things go and measure off our colors in here. And you can see that we end up with these lovely, lovely pictures like this. Hey, this is our sequence data right here. So if I were mean, I'd say interpret this sequence data. I'm not going to be that mean. But it's just kind of figuring out how these nucleotides are incorporated. And again, we can see our nucleotides are tagged with reporters, basically. All right, so there's our Human Genome Project, which is old news by now. We've figured out the human genome. We've moved on to the brain center research now. But we can figure out the human genome. And of course, the guy who did it sequenced his own genome because, we scientists are all narcissists at heart. But basically we can figure out different haplotypes. We can understand differences between our related organisms, like chimpanzees, something like we're like 99.7% identical to chimpanzee DNA. We can look at phylogenetic trees. We can look at the history of the human race and how they've migrated over time. See when cultures have interacted with each other. All that good stuff in there. For me, I'm most interested in identifying genes that are involved in disease because I like the whole idea of medicine and fixing people and making them better. Too much grew up with a $6 million man, Steve Austin. We can rebuild him and make him better. Instead of adding in cybernetics, we can just genetically modify someone, or we can do both. Anyway, away from science fiction, back to the actual science. These are the guys who did genomic sequencing. I actually met them. They're at a conference one time. I think I met Venter, and they're people. Anyway, our human genome has lots of different DNA in it. If we look at this, there are tons of introns in there. There are centromeres, long repeat sequences, simple repeats, short tandem repeats, all that stuff in there. Transposons, retrotransposons, things called signs and lines that we don't truly all sorts of stuff in our DNA. But only about 1.5% of the DNA codes for proteins. All right. So I don't know if you guys remember back when you like 1%, the whole Occupy Wall Street 1% and all that, and somebody put like milk in there. It's like 2% of milk in, or if you occupy the refrigerator. I thought I'd be cool and clever and make my own meme and say 1% of the DNA controls 99% of the protein expression. Occupy the chromosome. I thought that was a clever little meme. Thought it would go viral. I'd be cool on the internet and get lots of internet points, and 20 people liked it. It did not go viral. So, I guess, it was too smart for the general population. I say that on election day, given the candidates that we have to choose from. Yeah, I'm an elitist bastard, and I take pride in that. But good Lord. So that's why I push hard in this class, because you are the next generation and you need to be smarter and you need to be an example to the people in your community, wherever you end up, about how to learn, think scientifically, logically, and understand and investigate and make good decisions so that we can have a better society, instead of having, let's keep up with the Kardashians, or, what's on Instagram. So sorry, getting off track. Crush the old man. Get off my lawn. Anyway, but this is our nucleic acid makeup. There's really only this tiny percentage that's involved in expression, and the rest of it, we talk about junk DNA, but it's not really. It's kind of protective. You think about how many mutations are made. Well, statistically, the mutations are going to be made in something that is not too critical, like an intron or these signs or lines or these, all sorts of stuff that's out here. So, as long as we're not targeting a protein, odds are we're not going to make a serious mutation that will cause a disease. And even in the protein, there's enough variability that oftentimes mutations don't cause a disease. We get silent mutations that way. All right. If we look at just those proteins, what are our protein coding enzymes? Like 16% of them are, sorry, protein coding genes, 16% of them are enzymes. About 3% are in the cytoskeleton or extracellular structures, another 3% here, transport, regulation, signaling. And hey, if you want a career in science, more than a third of all the proteins are unknown what their function is. So there you go. You can figure out what the function of a particular protein is, and there's your career. Woo! Yay! Get your PhD, be a research scientist, or whatever. Sorry, I'm bitter, jaded. Yeah. Anyway, okay. So let's talk about single nucleotide polymorphisms a little bit. These are mutations that have occurred that maybe we have these different chromosomes from different people, and there's this little single nucleotide polymorphism. Something changes here. Individual 3 has a T instead of a C in there, or here we have an A instead of a G. Okay, so we can start accumulating these over time. We can come up with different haplotypes. Okay, so our haplotype 1 at these sites of polymorphisms would have an ATC sequence. Haplotype 2 would have an ACG sequence. Haplotype 3 has a GTC sequence. And haplotype 4, so we can see this over time. All right, so we can tag these different single nucleotide polymorphisms. If we keep track of lots of these, we can track the evolution of peoples over long distances of time. Okay, this can allow us to identify genes involved in diseases. If we look at like the family history, you may hear a lot about Ashkenazi Jews in biochemistry and disease, genetic and variant diseases in there. It's because this was a fairly insular people. No one outside was allowed to marry in for centuries. So they had a relatively uniform genetic code. So if you saw mutations in there, it was easier to associate these mutations with particular diseases. Closer to home, there is a family, or a series of families, all kind of distantly related in Kentucky, that there are the blue people. Their skin is actually blue due to a polymorphism, a particular mutation in there that will turn their skin blue. And so we can identify that and kind of figure that out. So, and a larger, more diverse population is a little bit harder because you're going to see a lot more of these mutations, a lot of polymorphisms that have occurred. So it's a little harder to narrow them down, but with enough data, you can narrow them down and figure out what genes are involved in particular diseases or well, I talk about diseases too much, but different mutations that may have beneficial or non-beneficial effects. Deleterious, I suppose I should say. All right. We can also look at the relationship between humans and chimpanzees and bonobos. Okay. If we look at our human chromosome 2, we can see that actually in chimpanzees, instead of having one single chromosome, they have chromosome 2p and 2q, but there's a lot of similarity on there, okay, as well too. We can look at these polymorphisms and see, how things have differed in there. And so basically we can construct a phylogenetic tree to look at how closely these are oriented. I suspect that this major shift in the chromosome also plays a big role as well as the individual mutations in here as well too. You look at chimpanzees and bonobos. I don't know if you guys are familiar with bonobos, but they are mean little monkeys. They they have a they like to yeah. You should just look up about bonobos, but they're very, very interesting. They're cute little monkeys for us, but they have some cultural activities that are very, very entertaining. That's all I'll say. Hopefully, I'll pique your interest and you can look up bonobos in there. But they're closely related to chimpanzees as well too. All right. So, but again, there's even though the DNA is remarkably similar, the differences that there are allow the differences in the species in there. Okay. All right. So you might say, well, hey, why don't we do medical research on chimpanzees? We do, but because chimpanzees are so closely related to us, it's a lot harder to get access to, say, a chimpanzee than a rat or a pig or a mouse. So we find things that are closely related. Again, I think I've mentioned that a lot of like skin stuff will be, pig skin is very similar to human skin. So we will do a lot of research on pig skin that way. Mice, transgenic mice, so they breed quickly, so you can get multiple generations really, really quickly. So they're a little more convenient to work with, even though they're not always as good as at predicting what happens in humans as well. Um, but yeah, but there are facilities that take very close care of our closest ancestors, but they are involved in some experimentation. All right. We can also figure out changes within human lineage. We can look at our genes in here. I always like this Gene X. It's the X-Men, right? Charles Xavier and superheroes. It's all just a fantasy. All right. But we can figure out common ancestors. Okay, so we can figure out where's our common ancestor. Where did we kind of divide off of here? Okay. So we can look at these different regulatory sequences, compensatory mutations that have occurred, and non-compensatory mutations, things that are making up for something and things that are just happen to have occurred. It's funny because I think about this in terms of looking at the iron sulfur protein. Almost everything has this one particular gene in there, one particular protein, and in the species I was looking at, it was serine. It was different, so I had to explain in all my papers. It's like, yeah. This is just a non, just some random mutation that has no effect on here, but that's why there's a serine here instead of an alanine or something or whatever it was. Um, Anyway, if we look at haplotypes, we can figure out migration. So if we start with the origin of the species, and we can figure out where different groups migrated, you talk about the different migrations. And one of the things that I've learned about in recent years is, say, the Han migration out of China. And you can kind of see, I think that's this M4 sequence running down through here, through Indonesia, yeah, something like that. But it's just always kind of interesting to track down, you know, how people have traveled in history. And we think about, was it Neanderthals and the what's the other really short, I can't remember the name for them, but there were other humanoid species there that Homo sapiens kind of competed with, and in some cases interbred with, and in some cases probably conducted warfare on, until we are the winners. The Homo sapiens, we're the winners, yay! But there's a fair amount of Neanderthal DNA in our DNA, maybe as much as 6%. And then was it the I can't remember the other. And and there are different groups that may have encountered these other humanoid systems that will have some of that DNA in there as well too. So we can just kind of use these different haplotypes to kind of figure out where people have moved to. And I guess was this M343. I guess if you live in Spain, yeah. Anyway, anywho, yeah, so humans and Neanderthals had a common ancestor some 700,000 years ago. So it's a lot harder for us to look at evolutionary data because it takes like 20 some years for each generation to occur. Compare that to, say, like E. coli, and you got 20 minutes and you got a new generation. Okay, well, maybe 30. So we can look at this kind of lovely little background in here. Here's our common ancestor 700,000 years ago. We can see this split off in between the two, and maybe like 8,000 years ago is when we and Neanderthals, well, when we beat them, we win, yay! Okay, all right. Oops, come on. All right, so that brings us to the end of this chapter. Very kind of all over the place chapter, I feel like, but the big thing in here is we're talking about recombinant DNA cloning, PCR, analyzing our DNA by sequence, mutating and amplifying DNA in a test tube, and then our expression vectors in here, and then figuring out our expression levels. So, we don't really talk about the haplotypes too much here, or the human genomic sequencing and all that good stuff in here. But just big takeaways here are some of these techniques. Our recombinant DNA cloning, our expression vectors, our polymerase chain reaction, and our chip arrays are probably our biggest things in there too. Maybe a little bit on the sequencing. Okay, so this is where I usually have some very open-ended questions on my exam. I'm trying to figure out how this is going to be probably the hardest exam for me to develop because it's usually very open-ended and free form, and there's a lot of choices on what you would answer in there. And I had to figure out how to do that in terms of on a Canvas exam. But speaking of exams, what I'd like to do is have our next exam next Thursday. Okay, so that means we're going to have some new material coming up. But this, I think, will give you a little bit of time, especially with the difficulty that we've had meeting last week. But this will give you guys a little bit of time to study, to kind of look at this and come up with questions. I will say that there is a wonderful study guide that is posted. Let's put that up here. All right. Yep. So here's our study guide posted in under the modules as well too. There's the homework in here that should relate to all this. But there's not there's not a bunch of crazy stuff coming out of this chapter. It's fairly straightforward. So it's a little more complex than the lipids and carbohydrates exam, but usually this exam is pretty good as well too. And I think the exam average on the last exam was like either like 78, 79, 81, 82, somewhere in that range. So somewhere close to a B minus, basically. So that's good. That's very good. This one's usually in the upper 70s as well too, I believe, average overall. Okay, but just kind of take a look at that. Start working through the homework. If you have questions, let me know. We'll do some questions on Thursday as well. I'll probably try and summarize this and go over the study guide a little bit. Wow, I talked a lot faster than I expected. So let me ask you guys now. Do you have any questions now? Yeah, now you're just kind of taking it all in, right? And you can type them in the chat too if you want.
Dr. Ebert,
Yes.
where do you post the video recordings? Because I couldn't find them in the modules.
Yes. Okay. So they have their own module up here. There is something called Zoom recordings, and I kind of named them by their chapters. So for example, the exam 4 stuff will be start with 8A, 8B, and 9A is the one I made there, and then today will be 9B. Okay. But they should be right underneath the start here module. There should be something called Zoom recordings.
Okay, thank you.
You're welcome. Oh, and of course, I'm looking over this. I didn't share my screen, so let's do that. Yeah. Okay. There we go. So if we go down here under modules, and I really like modules. I think the hardest thing is I think in terms of modules, and a lot of students don't. But the second module is called Zoom recordings. So if you scroll down here, you'll find them, and today's will pop up here as well too. I'll put them in order, and I try and indent and put things my different lectures. That's really weird that I did it that way. Anyway, okay. I was gonna say 2 meetings down, 2 more to go today. My throat is gonna be so tired by the end of today. All right. So I got a question here about extra credit opportunities. That's an interesting question because I know being primarily online this semester, I had to look at how things, how scores work out. I will have some additional assignments that are coming up. I know in the next chapter, there, I think chapter 11 and chapter 12, I have 2 other assignments that will show up there. Sometimes I make those somewhat extra credit, as in there's a certain amount of credit, and then there's a bunch of points that become extra credit that will add into usually like the overall homework grade or something like that. I usually don't like to put in a whole lot of extra credit opportunities, but there are a few points here and there. I make sure on the upcoming exams that they're usually built in an extra 5 points. And I actually need to go back and fix that in some of the early exams because I didn't figure out quite how to do that. So for example, like a lot of the scores in like the first exam are out of like something like 105 points, well, it should be out of 100 points, and those extra 5 are the bonus. So you'll see scores will actually improve once I actually get back in and fix that. But there will be small built-in extra credit opportunities is what I guess is what I'm saying. Yeah.
I have a question.
Yes.
When is the final? Or you
Uh,
can let us know whenever. I just wanted to like
yes,
get an
yeah.
idea.
I will know after exam 4 when that is. My assumption is it'll probably be the Tuesday before Thanksgiving. Potentially, we've talked among the faculty about all classes end by Thanksgiving, but final exams will be online after Thanksgiving. I don't really like that idea, but I may leave it. It'll depend on how far we get in these next 2 chapters, how soon. I think we've got plenty of time, and to still have the exam like that. Was it November 21st or something like that? But I'll let you know as soon as I know. Probably within a week, I'll have a good idea of when that final will be.
Okay, thank you.
You're welcome. All right. Other than that, I guess I will stop recording.